oftentimes we have signal about the biology at the first initial event, but what happens afterwards has often been dark matter space. And I think the autopsy program has really been transformational for CBTN, particularly in partnership with Gift from a Child and under Javad's leadership, who needs her introduction and has been my partner in crime for a long time.
Thank you, Adam. Thank you very much. I cannot believe I'm talking after Aaron. This is so hard. You gave such a beautiful presentation. I'll try to make this quick then. And then the title of the talk was selected by Adam. Thank you very much. I found this out yesterday. I looked up the literature to see how many publications have been done on post-mortem brain, how many publications have been. If you look at the graph that we have here, from the PubMed publication in 2000, we've had 1,500 papers published. If you add the word post-mortem brain cancer, the numbers as of 2000 have been 126. If you add pediatrics, the numbers are much lower. As of 2000, there have been about 17 publications that have come out that describe pediatric post-mortem cancers that have been published. A few of those come from our work that have been supported by the Gift from a Child and the Swifty Foundation. And the reason that it's really difficult to do such post-mortem processing for pediatrics population is this diagram that again has been published by our colleagues, Vijay and our colleagues from the Swifty Foundation. It's a very complex process. It takes a lot of coordination between the site where the patient wants to donate. The donation is typically, the decision to donate is typically is made late towards the end of the patient's life. The coordination site is far from the patient where the patient is donating. And our colleagues from Children's National and from Swifty Foundation are here today, can talk to them. It's a very complicated and lengthy process. That's the reason I think we have not been too good in collecting patients post-mortem. But just to highlight our efforts at Children's National, the donations per year have been increased. This is post-mortem donations per patient, and you can see post-2019, the numbers have been going up. That's when Children's National became a center of excellence that was funded by the Swifty Foundation to increase such efforts. And it really takes the whole team to coordinate such post-mortem from patients. And donations total, you can see that they also have been increased. And a lot of the credit goes also to the clinicians who are telling the patients that there is an option for post-mortem donation. A lot of credit goes to families who have decided this really brave decision to donate their patients, their child's tumor at post-mortem. And here is a diagram of the sex and also the diagnosis of the tumor types that we are collecting. We collect all types of tumors so long as parents want to donate post-mortem. I mentioned the Swifty and the Gift from a Child. They have established multiple different sites across the US. We have actually worked with Swifty to collect post-mortem samples outside of the US as well, notably Canada and 2 sites across Europe that we have collected post-mortem. There are centers of excellence that are listed here that coordinate post-mortem connection and coordination across the US. I wanted to show an example of what we have done with the samples that we have collected. We decided to, as you know from the CBTN, many sites, many institutions usually put in a request to collect a sample. And for most of the time, they want to look at expression of the proteins that Aaron, for example, showed today. One effort that we wanted to have was to build a tissue microarray where we will put samples from all of the patients who have donated to us and to other sites within the coordination sites into one tissue microarray where one could ask CBTN to get just one cut of that tissue microarray and to look at different proteins that they have. The initial effort that we had, the patient numbers are listed here. We had 70 patient whole brain that we included in the tissue microarray. We also worked with the NIH and other institutions to get brains from patients who have died from non-cancer related causes. We collected 10 brains that we added in the tissue microarray. One of our colleagues, a neuropathologist in Switzerland, reviewed all of the slides. This was a hefty work. I think we had over 1,200 slides that were reviewed by the neuropathologist, and she demarcated the areas where the tumor exists, as you can see here on the slide. The tissue block was subsequently taken to get the punch cords. You can actually see where the punches were taken from that block and put together to create a tissue microarray that has now over 900 cores. These are 900 cores that represent 3 different sites from each patient. And this is the primary tumor site, metastatic site, and an adjacent healthy. This is the largest tissue microarray that has ever been created for pediatric brain tumors. Then the question was that we asked the pathologists, if you want to cut this tissue microarray, how many times can you cut this before it's exhausted? The pathologist said that we could probably cut this 40 to 45 times. And we asked our working group, what are the antibodies that you guys want us to stain this with? And we had 450 different antibodies. So it was almost impossible to use these to stain all of the samples that we wanted. For that, we were introduced to General Electric of all the places. They have a system called CellDive, MXIF platform, where you can take one slide and essentially stain it hundreds of different times. So long as you have the antibodies that you can put 4 antibodies at the same time to restain it, essentially one slide would be enough to stain for all of the antibodies that we wanted. You will keep doing this until the tissue will fall off. This was the work that we done with them that took about 1.5 years. We screened over 80 different antibodies. We successfully stained 45 antibodies. And we compared this to the staining that was done by the neuropathologist. And the IHC immunohisto, you can see the actual staining, the traditional way, where you take the tissue microarray and you stain it with one antibody. In the bottom, you have CellDive, where it is immunofluorescent. You stain, you kill the signal, you stain again. And we're looking at the concordance. The graph that you see on the right is looking at the pons, cerebellum, thalamus, and cortex of a patient, and counting the number of positive cells in the IF compared to the IHC. And you can see that they're very comparable. So essentially, we have developed a platform where we can do multiple antibody staining across the 900 plus samples that we've had. Then we included the antibodies that our colleague wanted, specifically antibodies that are important for immunotherapy and immunologists. And you can see here the concentration, the density of CD3 positivity, CD68, and B7H3 across different tumors, primary site, metastatic, and tumor types. Noting that the majority of the samples that are on this tissue microarray are DMGs and DIPGs. And then we started correlating, looking at the PD-1 expression, PD-L1 expression. The blue, the green light here is DIPG. Is it green or blue? Aqua. Aqua. The aqua one here is DIPG. You compare it to the healthy. You can see that DIPGs are highly dense with PD-1 and PD-L1 expression. And you can actually see the tumors that are expressing K27M positivity in the context of a particular marker for a particular immune cell. We have mapped the immune landscape across patients that have been treated here. You can see patients that are treated with ONC201, for example. In the primary DMG, you have a higher concentration of CD8 positivity in some of these patients. You can also see patients who have received an immunotherapy. This is the Newcastle virus treatment. Some of you remember this was done a few years ago. Patients who had received Newcastle virus treatment, and a few of them had received also early versions of ONC201. You can see actually an activation of the immune system within their tumor types. Some of the analysis that I'm showing here were done by our colleague Andrea, who will have also a talk tonight. And we have seen the work of the utility of the postmortem data, again, for particular response to a drug, in this case, ONC201, that was done by Carl and Sriram. Sriram in the bottom, who looks so excited to looking at the Western blot. I've never seen anybody to be so excited to look at the Western blot. They showed that patients who received ONC201, there's a higher expression of K27M trimethylation. Again, utility of the postmortem samples that we have received. And then Of course, we wanted to take the data that we have generated, again, 900 plus cores that have been stained with 40 plus antibodies. We work with Sarah, who has been working also with CBTN. She gave a talk at the Pete Snow a few months ago here in Washington. She took all of the data that we have and developed a machine learning algorithm that can now take any naked tissue, a tissue that has not been stained, and she can predict the expression of 14 different antibodies across any samples. And she has now a student who has come here to Children's National to work with CBTN and to work with us to look at the samples that we have to collect the images that then she can increase this AI system that we have. Our hope is really to develop a platform that people can upload naked pictures of the tissue that they have, and we can use the platform that we have here to detect and to predict what are the antigens that are being expressed. And of course, all of the data will be freely available and shared through the CBTN platforms to all of our colleagues who want to look at the expression of the proteins. But what was really interesting, a surprise that came up to us, was the expression of B7H3. And I know colleagues that we have here, Dalia and others, have published on B7H3, or they have an interest in immunotherapies that are involved in B7H3. When we look at our patient cohort, you can see expression of B7H3 in the tumors, but you can also see expression of B7H3 in the healthy cells. And the question was that who or which cells are expressing B7H3? If you look at this image that we have generated here, B7H3 expression does not co-localize with K27 impulsive cells. There are very few DIPG cells that are expressing B7H3. Other than that, we have not been able to look at the B7H3 expression. And you can see here again, in a higher magnification, you have a DIPG cell that is expressing B7H3, but essentially most of the cells that we detect, they don't have B7H3 expression. And the question was that because these are post-mortem samples, is it possible that our post-mortem samples have changed at initial diagnosis? You would have more expression of B7H3. We're working with our colleagues at Princess Maxima in Europe, in Netherlands, that have also seen similar trend that they cannot really detect B7H3 expression. A few months ago at the meeting here, the Pete Snow meeting, we discussed this with our colleagues at Hopkins, and we told them that if we send you our tissue microarray, would you stain it with B7H3 and run it by your pathologist to see if you observe the same thing? This is data that came from Hopkins last week. They can also see that primary metastatic and adjacent healthy, they do have expression of B7H3, but it's not as pronounced as what we expected. We then took the B7H3 sample, we then took the post-mortem samples, and this work done by Sridevi. We did a Western blot analysis to see, do we detect them, at least with the Western blot analysis. And what you can see here again, adjacent healthy do have an expression of B7H3. Tumors also have expression. So this is a discussion that we should have as a scientific community, specifically using antibodies that we want to take into the clinic for immunotherapy. What is the expression of a protein? And those of you who work with CBTN data, you know that the expression of RNA almost never or barely correlates with the protein expression. The expression that we see at diagnosis is very different at the postmortem. But the postmortem space gives us this opportunity to ask these questions and to answer them. Again, this is the RNA expression across all of the DMG cell lines. If you look at the cell lines, they all highly express B7H3, but not the postmortem tissue from the patients that we have looked at. We've also utilized the postmortem samples that we have. And again, this is the work that's being done by Aaron and Masahiro at UCSF. They have looked at the postmortem samples from patients who had received ONC201. And they've asked the question, what is different about this patient population compared to other patients who did not receive ONC201? And they have actually mapped a very novel pathway, a BDNF pathway, BDNF1 pathway that has been known to be involved in DIPG growth. And their work has shown that there's an activation of this pathway that potentially creates the resistance of the DIPG cells to ONC201. We are working very closely with the Swifty. with other institutions and with CBTN to build a tissue microarray 2.0. Caroline, our colleague from Children's National, is here. If you're interested to join these efforts, please reach out to us. We will include tumors across all ages and all types, so long as they're CNS tumors. And we will be working with GE for spatial mapping, but also RNA mapping across all of the tissue microarray. If you're interested, please join us. Thank you.
I think this concludes our scientific presentations for the morning. What you can, at least what I feel, right, is this amazing complexity, right, that's emerging across these large-scale data sets, across modalities, across approaches, the required architecture. Ash highlighted, even thinking about clinical data and intersection imaging data. That's both the challenge and opportunity and excitement, at least that feeds, I think, almost all of us here in the room day to day. Before we close, two things. One, I want to remind you that we just released almost double, right, the amount of data. All of that is available within the Kids First portal at 12:10 when we break for lunch. There will be a training session at the Skills Center for all of you to engage, play, learn, about how to use a lot of that data, as well as the GFAC working group.